So if we look at some of the detailed accounting, um, so what we have is the ATP directly made by the pathways. This is actually via substrate level phosphorylation. And this is in this whole table is about the ATP yield per glucose. So in glycolysis for 1 glucose, we get 2 ATP net made via substrate level phosphorylation. I'm going to remind you that glycolysis yields 2 pyruvic acid, and then that means 2 acetyl-CoA will be entering the citric acid cycle. So every time the citric acid cycle proceeds, we get 1 ATP per turn. So if it's going twice, we get net 2 ATP from 1 glucose. So we get net 4 ATP from substrate level phosphorylation under aerobic conditions. Under anaerobic conditions, we only get the 2 because there is no citric acid cycle occurring under anaerobic conditions. It would only be the 2 ATP via substrate level phosphorylation. So if we look at the ATP made via oxidative phosphorylation, let's just focus on the actual yield. Okay, so for the yield from oxidative phosphorylation, we can get up to a total of 5 ATP because you get each NADH plus H is going to give 2.5 ATP in the electron transport system. So 2.5 times 2 is going to give 5 ATP from 1 glucose. If we look at the conversion of pyruvate to acetyl-CoA, we know that there's the 1 NADH plus H generated per pyruvate, and that happens twice. So we get net 2 NADH plus H. That's going to again yield the 5 ATP, that step going from pyruvate to acetyl-CoA. And then if we look at the citric acid cycle, right, that's going to occur 2 times per glucose because there's 2 pyruvate coming in, converted to 2 acetyl-CoA. And then we're going to get, there's going to be a net 3 per cycle, NADH plus H, 1 FADH2 per cycle, but this is going to occur twice, right? So then we're going to have 6 NADH plus H times 2.5, giving 15 ATP, and then you get 2 FADH2, so 1.5 times 2 gives you 3 ATP. So you get 18 ATP in the citric acid cycle from oxidative phosphorylation, 5 in the step in between glycolysis and the citric acid cycle, and then you get up to 5 in the glycolysis. So basically, you can get about 28 ATP, right, from oxidative phosphorylation, in contrast to the 4 ATP you get under substrate phosphorylation in the presence of oxygen. In the absence of oxygen, you only get 2 ATP. So the last things I wanted to talk about are the aerobic metabolism specifically of lipids and proteins. And we all know that from what I told you before, that if you eat excess carbohydrate, you can store that as glycogen. So that's a really good thing that endurance athletes do. If you eat a lot of carbs the night before a big race, again, you can store that excess glucose as glycogen. If you eat too much food, we know, and if you're not an endurance athlete, you can actually have your excess carbohydrates converted to fat. And so basically here, this is how 3-phosphoglyceraldehyde can be converted to glycerol. Acetyl-CoA can be converted to fatty acids, and then these can be combined together to make the triacylglyceride that is a common form of fat in the human body. So one of the reasons that exercise helps you to lose weight and not put on extra weight is that aerobic exercise favors your metabolism to be producing more glycogen instead of fat when you do eat a high carb meal. So, but in any case, we all have some fat, and how does the body metabolize that fat? So a triacylglyceride we'll have to have the fatty acids removed from the glycerol backbone. And once that happens, the fatty acids can be moved to the mitochondria, where a process of beta oxidation can occur. So here on the top, we see a fatty acid. CoA is going to condense in here. The fatty acid has to be attached to CoA in a condensation reaction. And ATP is going to be put in, right? And that's putting energy into the system. So this is a endergonic step, right? Adding ATP to the system because you needed energy for this to happen. And then we're going to see a redox reaction. So FAD is going to be reduced to FADH2. And that means this fatty acid has been oxidized. You can see it oxidized because now there's a double bond being formed. There's a hydration reaction. There's going to be another redox reaction where this molecule is going to be oxidized, and NAD is going to be reduced to NADH plus H. And then what happens is there's going to be a cleavage event where the 2-carbon unit will be cleaved off from the rest of the fatty acid, and it's always the carbons are always cleaved off in 2-carbon subunits. So this 2-carbon group is going to be an acetyl group. So acetyl-CoA will leave this fatty acid and then can enter the citric acid cycle. So in the citric acid cycle, each acetyl-CoA can yield a total of 10 ATP via substrate level and oxidative phosphorylation. And then you also can get, via the electron transport system, FADH2 generating 1.5 ATP and NADH plus H generating 2.5 ATP. And remember, each of these steps is happening for every 2 carbons. So if you have like a 16-carbon fatty acid, you can do this 8 times, right? So basically, 1 molecule of a fatty acid can give you way more ATP under aerobic conditions than 1 molecule of glucose. So fat is a really great source of energy and ATP. And again, this is occurring, beta oxidation occurs inside the mitochondria. So the last thing I just wanted to mention is the conversion of amino acids to other amino acids. And a lot of amino acids are generated from other amino acids by transamination reactions. So amine refers to these nitrogen groups. So here what we're going to see is the NH2 group is going to, from glutamic acid, is going to be added to the this carbon of oxaloacetic acid, and in this transamination reaction is going to generate alpha-ketogluteric acid and aspartic acid, right? You can see that what happened here is that the NH2 group was moved, and also this oxygen was swapped to that carbon over here. So 2 things happen there critically, and then so what you can see is you have these 2 amino acids, and guess what? Another 2 amino acids can be generated by this transamination reaction. Another example is with a transamination reaction involving glutamic acid and pyruvic acid. Again, we're going to see is the transfer of the NH2 group over to this carbon, and then the oxygen is going to be transferred over here, and this will generate alpha-ketogluteric acid and alanine. So transamination reactions really allow our cells to generate the different amino acids that it needs if you haven't ingested them by your digestive system. So the pretty interesting thing is that protein is a great source of nutrients too, and proteins, when they're hydrolyzed to amino acids can be converted to the citric acid cycle intermediates. Intermediates are just like the molecules of the citric acid cycle. You don't need to memorize which Yeah, so the different amino acids can be converted into molecules in the citric acid cycle. So you don't need to memorize which amino acids are converted to which citric acid cycle molecule, but I just want to leave you with this appreciation. So, for example, alanine, cysteine, glycine, serine, all these amino acids can be converted to pyruvic acid. And during this conversion, there's going to be a transamination reaction, which will lead to urea being secreted, and that usually ends up in the urine as a waste product. Leucine, tryptophan, isoleucine can be converted to acetyl-CoA, right? Arginine, glutamine, histidine, proline can be converted to alpha-ketogluteric acid, so et cetera, et cetera. But all these amino acids can funnel into the molecules of the citric acid cycle. So this is how amino acids can generate ATP. Yes, they don't go through glycolysis, but we know that the citric acid cycle can generate 10 ATP for each time it goes around, right? So amino acids are also a great source of energy. So this concludes our cellular metabolism lecture, and I wish you all a great day, and take care. Bye-bye.